Scientists have recreated entire human organs in a dish, and the most astonishing breakthrough hides at number 1. Stay tuned to see the organoid no one expected to dominate the list. Imagine a tiny liver that can detoxify chemicals, process nutrients, and even predict drug toxicity, all without a single animal. Researchers cultivate hepatocytes in a 3-dimensional scaffold, allowing them to form bile duct structures and metabolic zones that closely mirror a real organ. This miniature liver can flag hepatotoxic side effects weeks before a compound reaches clinical trials, saving both time and resources. Moreover, patient-derived liver organoids let scientists study genetic liver diseases in a personalized manner, opening doors to targeted therapies. As the list climbs, a beating heart in a petri dish awaits, showcasing the next level of complexity.
Remarkably, kidney organoids now mimic filtration and reabsorption functions that were once exclusive to living mammals. By guiding stem cells through precise signaling pathways, scientists coax them into forming nephron-like units complete with glomeruli and tubules. These mini-kidneys can model acute kidney injury and test nephrotoxic drugs, providing a humane alternative to traditional rodent studies. Some teams have even connected organoids to microfluidic chips, creating a flowing blood-like environment that enhances physiological relevance. The next segment shines a light on a mini-brain that thinks beyond expectations, pushing the frontier of neural research.
Surprisingly, pancreas organoids can generate insulin-producing beta cells that respond to glucose swings much like their in-body counterparts. By mimicking the developmental cues of the pancreatic bud, researchers coax stem cells into forming islet-like clusters that secrete insulin in a regulated fashion. This breakthrough offers a dual advantage: a testing platform for diabetes drugs and a potential source for cell replacement therapies. Early trials have shown that transplanted organoid-derived beta cells can lower blood sugar in animal models, hinting at future clinical applications. Up next, a breathing lung will take your breath away, demonstrating how organoids can emulate gas exchange.
Hidden within a glass dish, a beating heart organoid contracts rhythmically, echoing the pulse of a living organ. By arranging cardiac progenitors into spheroids, scientists achieve spontaneous electrical activity and coordinated muscle contraction. These miniature hearts can be paced, stressed, and monitored for arrhythmias, providing a powerful platform for cardiotoxicity screening. Moreover, patient-specific cardiac organoids reveal how genetic mutations alter contractility, informing precision medicine approaches for heart disease. After this, an intestinal organoid that mimics digestion will appear, showcasing another layer of bodily function.
Few realize that gut organoids now recreate the complex architecture of the small intestine, complete with villi, crypts, and a functional barrier. By exposing stem cells to WNT and Notch signals, researchers generate self-organizing tubes that absorb nutrients and host a microbiome-like community. These mini-intestines enable studies of nutrient uptake, inflammatory bowel disease, and even viral entry without animal models. They also serve as a testing ground for oral drug formulations, predicting absorption rates with unprecedented accuracy. Next, a skin organoid that heals wounds in minutes will surface, illustrating regenerative potential.
Deep within a petri dish, skin organoids form layered epidermis and dermis that respond to injury just like real skin. By coaxing keratinocytes and fibroblasts to self-assemble, scientists produce a living tissue that can be wounded, repaired, and even pigmented. This model has accelerated testing of cosmetics, scar reduction therapies, and gene editing approaches for conditions such as epidermolysis bullosa. In some studies, engineered skin organoids have closed microscopic gaps within hours, hinting at future applications in burn treatment. Up next, a retina organoid that sees light like real eyes will illuminate the list.
Unbelievable! Retina organoids develop photoreceptor cells that organize into layered structures capable of responding to light stimuli. By guiding pluripotent stem cells through ocular development pathways, researchers produce mini eyes that generate electrical signals when exposed to photons. These organoids have been used to model macular degeneration, test gene therapies, and screen neuroprotective drugs, all without sacrificing animals. Some teams have even connected retina organoids to microelectrode arrays, capturing real-time visual responses. Now, the ultimate multi-organ chip is about to unfold, promising a holistic view of human physiology.
Astoundingly, cerebral organoids, often called mini-brains, grow layered neuronal networks that exhibit early brain-like activity. By allowing stem cells to self-organize in a spinning bioreactor, researchers generate structures with cortical regions, synaptic connections, and spontaneous electrical bursts. These organoids have modeled neurodevelopmental disorders such as autism and Zika-induced microcephaly, offering insights that were previously inaccessible. Moreover, drug screening on brain organoids can reveal neurotoxicity before animal testing, reducing ethical concerns. Stay with us for the final marvel that integrates many organs into a single platform, redefining drug discovery.
Incredible, lung organoids now form alveolar sacs that exchange gases, mimicking breathing at a microscopic scale. By differentiating stem cells into airway and alveolar epithelial cells, scientists create branching structures that produce surfactant and respond to pollutants. These models have accelerated research into COVID-19 infection mechanisms, asthma therapies, and inhaled drug delivery, providing a human-relevant alternative to rodent lungs. Some lung organoids even integrate vascular endothelial cells, allowing fluid flow that simulates blood-air barrier dynamics. And finally, the number one breakthrough will change everything.
Finally, the body-on-a-chip unites liver, heart, kidney, brain, and more into a single microengineered platform, letting scientists observe how drugs travel through and affect multiple organ systems in real time. This interconnected network of organoids is perfused by microfluidic channels that deliver nutrients and waste removal, recreating the circulatory system without a living animal. Researchers have used it to predict human drug responses, identify toxicities, and explore disease pathways that involve organ-organ crosstalk, such as diabetes-related kidney damage. Reportedly, this technology could reduce animal testing by up to 80% and shorten the path from discovery to clinic, marking a paradigm shift in biomedical research.
What a journey through the living laboratories of tomorrow. From single-tissue marvels that detoxify and contract to the integrated body-on-a-chip that could replace whole animal studies, these organoids are rewriting how we discover medicines. If you loved this countdown, hit the like button and subscribe so you never miss the next mind-blowing list, new discoveries every week. Which organoid left you in awe, and what would you like us to explore next? Share your thoughts below, and thanks for watching.